Okay, so starting on page 6, we'll do the using simplified diagrams show how glucose molecules join to form disaccharide maltose and the polysaccharide starch. So this is going to be through a condensation reaction. Condensation reactions are anabolic, meaning that energy is required. And so we'll first start with drawing in our glucoses. It's a starch, so they're going to be alpha glucoses. So we have 1, let me just draw in the little structures for both first. Okay. And so I'm going to draw in the molecules that will eventually become our water in a different color just to help you see them. And so this one will give our OH and this one will give our H. So we have O, H, and we have OH. But remember that they're bonded to these molecules. Then we'll draw in the rest. OH, H, OH, H, OH, H, H, CH2OH, H, CH2OH, H, OH, OH, H. So that's step 1. We just have our molecules, and then energy, or like ATP is added. So ATP. And so then step 2, unfortunately you have to draw in your glucoses again. So O, again, let me draw in the structure first, blah, 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 blah. Boop. And then H, OH, OH, H, H, CH2OH, H, CH2OH, H, OH, OH, H, H, OH. Oh my goodness. And then at this point, these that were originally bonded to these two molecules, because of the addition of ATP, they're going to kind of separate off to be an O with 2 H's. And oh my gosh, look at what we have when we have an O with 2 H's. It's H2O, which is water, and that leaves. That goes and does whatever water does in your cell. It's like a byproduct, it's like waste. And so then the final step, step 3, is that they form the bond that actually links these two monomers together. So once again, it's time to draw your glucoses. So O, H, OH, OH, H, H, CH2OH, H, OH, H, boop, boop, boop. And then now we have this O down here from the other one, but they're going to form a bond because of the removal of the water. So we have OH, H, H, OH, H, OH, H, CH2OH. And these bonds that form are your glycosidic bonds. And they will be what links all of your carbohydrate monomers and polymers. And then we also have plus our O, 2 H's, or our water, which is now just like its own separate thing. And so we'll label everything else. Oh, let me get a different color for the labels. We'll make it fun. So this new molecule down here is maltose, which is C12H22O11, because we lost the 1O. And then these are both, oh, how am I going to make this arrow work? Glucose, which are C6H12O6. Boop. Okay. And let me draw a little line between these. And we'll do our polysaccharide forming, which, forgive me, even in my draft, my polysaccharide formation drawing was very questionable, so forgive me. And so polysaccharides, again, it's more than 2, it's many. I am not going to put you through drawing all of those glucoses, but they are all glucoses. And so just to make it simple, step 1, we'll have our implied glucoses. We'll do 3. That way it's more than 2. And they're all going to be alpha glucoses because it's starch. We'll make this one be the one that gives up the H. So this is your starch. And then you'll have another glucose. It can either be a monomer, or it can be like a couple of them bonded together. It doesn't really matter. All of them are going to bond in about the same way. For the sake of simplicity, we'll just do 1. Because we drew them all the way out, and the other ones I'm going to be lazy and not draw them all the way out. So we know this is a glucose. We can label it. This is glucose. It's specifically alpha glucose. And it's going to have an OH. Same thing happens. We have ATP. Ooh, we'll draw a little arrow down. We have these guys in blue go off and become H2O. And then our end result is, I guess this arrow should be black then, huh? Is our 4 starches bonded together, or our 4 glucoses bonded together to be one nice happy starch. 2, 3, and then we'll show our little glycosidic bond. H, O, oh, I forgot to put the H up here. H, and then our other glucose. But they're all bonded together the same way. This is just my like lazy way of showing you the bond still. So it'll still form the same bond. They all technically still have this little O bond between them. It's just implied. So now it's just one big starch. One long starch. You can draw in all the O's if you want to between all of them, but again, I'm being lazy. All right, on to the next page. Bum-ba-da-dum. So, first question, disaccharides, which are 2 monomers, and polysaccharides, which are many, are formed when monosaccharides join together in a condensation reaction. What exactly is a condensation reaction, and what byproduct is released? So what we just modeled is a condensation reaction. And so, ooh, what color do I want? That's fine. So, condensation reactions are anabolic, meaning that they require energy, you can think that the energy is ATP, anabolic starts with an A, if that helps you. So condensation reactions are anabolic reactions whoops, let me flip my page over, which join 2 or more monomers together. Water is released as a byproduct. I'm just now realizing that that color is really hard to see. Oh well, it's too late. I've already done it. I'll pick a different color for the bond type. And so, what is the name of the bond that's formed between 2 monosaccharides? It is a glycosidic bond. Woo! And so the opposite of condensation is hydrolysis. And so hydrolysis is when you add water and remove energy to split 2 monomers, or 2 polymers, one polymer into 2 monomers. So you have 1 polymer, add your water, split 2 monomers. And that water is also split, so that you replace the OH group and the lost H that we lose in the condensation reaction. And so now it's time to put that into words in not that color. I feel like writing it in red's aggressive. I had a green. There we go. So, hydrolysis is a catabolic reaction. Catabolic means that energy is released. I like to think cats are like lazy, and so they don't need additional energy. I don't know. is a catabolic reaction where water is added to split polymers into monomers. The water is also split to replace the hydrogen and OH to stabilize the monomers. And so you see your water here, and then you see the OH and the H get added back on. I feel like that's hard to see. Where's the highlighter? You have your water here, you see the OH and the H get added back on. So they have a little compare and contrast. So you have your polymer, your monomer, water is removed to have them join together. You have your polymer, water is added to split them. And so in this case, start with the water, it goes away, they come together in condensation. In hydrolysis, don't have the water is added, and then it's split to be our OH and our H. I feel like these arrows are not super obvious. I'm going to make them nice and thick. It goes away. It goes back in. And then here you'd have your, let me get a different color. Glycosidic bond. And then in this case it would be here. They're basically like reverses of each other. They're essentially like opposites. And so in the little table, so differences. These are also hard to look at. Okay, well, we'll do condensation in red. Condensation. It's anabolic, meaning that energy is added. Water is removed. And it goes from monomers to polymers. Hydrolysis is catabolic. Energy is released. Water is added. You could think it's rehydrating, it's hydrolysis. Lysis also means to split. So it's splitting with water. Condensation, you're condensing 2 to 1. It's going from polymers to monomers. And then we'll write the things that are the same in purple. So similarities, both involve water. Both involve energy. Both change. Monomers / polymers. Next page. So now we're going to look at the different polymers individually. So we have our starches. Where are the highlighters? There they are. Okay. So for starches, again, we have two different types. We have amylose, and then we have amylopectin. They are slightly different. There are some things that are the same about them or consistent. So they are both in plant cells. They both use alpha glucose. And then the structure is where they start to kind of differ. Hang on, let me get my colors situated. So for amylose, it has unbranched chains that form this kind of curved helix. For amylopectin, it has branched chains, branched straight chains, that branch off about every 20 monomers or units. So about every 20 glucoses, it's going to branch. And remember, both of these, like all of these big polymers are all just glucose. And then for the types of bonds, for amylose, you have alpha 1,4 glycosidic bonds. And so that's just talking about where on the glucose it's going to bond. Remember, we went through and we numbered them. And then for amylopectin, you have alpha 1,6 glycosidic bonds. And so you can see that in your amylopectin. Oh, I guess I'll go to highlighter, that way it doesn't make it all grody. Here. And for your amylose, you can see them here. Your amylopectin is also going to have some 1,4 bonds, but those branches are going to come from your 1,6s. And then in terms of function, what color should function be? I want to write in red. We'll do functions in orange. And so for function, these are all mainly just storage of energy in cells. So storage for energy in cells. The key thing about it is that the glucose can be removed really easily. For things like cellular respiration to release that ATP, but the polysaccharides are less soluble because of how complex they are, making swelling from osmosis less likely. Which is like really a big deal, because if we have an unbalanced amount of sugars inside of the cell, water will rush in, and that can actually cause cells to burst and die, which is clearly an issue. Like, you want to be able to store energy for your cells, but you also don't want your cells to die because they're swelling with water. On to our next one, our glycogen. So plant or animal, it is in animals and fungi. Alpha or beta. Where's the stupid color? There we go. This one is also alpha glucose. This one only has one type itself, and it is branched, straight chains, but this one branches about every 10 units. Meaning it branches twice as much as amylopectin. More branches equals more complex. This one also uses a lot of your alpha 1 to 6 glycosidic bonds. Again, it has the 1 to 4s to keep the chains. I guess I should put that in too. And 1 to 4. But it makes those branches, which are like its key thing, with the 1 to 6s. So we have our 1 to 4s. And then our 1 to 6s. And then the function, it's similar. It's only a little bit different. Except for this one is storage for energy in animal cells. Again, the easy access to glucose. Glucose without swelling. This is pretty much insoluble because of how complex it is. It's made in muscle and liver cells, in humans at least, and most animals. Where it's made in fungi, I actually don't know. And then our last mystic journey, flippy flip flip, is cellulose. This is probably the most special friend of the bunch. It's the most different friend. It is in plant cells. Its biggest difference comes from the fact that it uses beta glucose rather than alpha glucose, which is part of what gives it its like different structure. And so what makes it so different from the other two and why it needs to use beta glucose is because not only does it have these chains that are formed by your beta glucose, but it also has hydrogen bonds that then form in like little ziggy-zaggies to take your chains and bind them into like sheets, which are then woven into these microfibrils. And so beta glucose is added to chains, parallel chains link with hydrogen bonds becoming bundled microfibrils. And so it has your beta glucose 1 to 4 glycosidic bonds, but then it also is special because why wouldn't it be? And it also has hydrogen bonds. And so let's mark where those bad boys are. So you have your 1 to 4s. Right? Oh, I guess these ones too. Okay. And then, bonus, you also get these hydrogen bonds that form these like little ziggy-zags for bonus stability and bonus structure. Look at them go. The function, they are in cell walls. They are super strong and rigid. They provide tensile strength, to the plant, cells, allowing them to not blow up under pressure. That's what hold the plant up. It's like a bonus fun fact. But blowing up cells based on either osmosis due to imbalance of like concentration of solute in water or because of like not enough barrier for the pressure is a common theme. Carbohydrates do a lot for that. And then our final piece, I think, right, for carbohydrates? Oh no, there's still the blood part. Our almost final piece for carbohydrates is comparing the structures of starch, and this is wrong, liar, cellulose. And so key things, starch has alpha glucose, cellulose has beta glucose. And then if you look at them, if you look at where you see your like loose OH groups with beta glucose bonds on the 1 to 4, they end up like on these opposite sides. If you look at where your loose OH groups are on your starch, they're all on the same side. Having them all on the same side is what allows for branching. Because it allows those 1,6 bonds to form from here, or from here to here. when you have your OH groups and your CH2OH groups on like catty corner sides, that's what allows those little diagonal hydrogen bonds to form. So the fact that we have both alpha and beta glucose and the fact that they're different is a key component to why the structure of these polysaccharides is the way it is and why they function the way they function. So allows formation of H bonds. All right, on to blood. I was gonna make you all do lipids, but I can see now that this video is like 30 minutes long, so I will save you all from doing the start of lipids too. Okay, time for blood. I promise blood will be fast. So blood, get excited. And so when you're looking at the outside of a cell, you have glycoproteins on membranes that have they're proteins that have a carbohydrate attached to them. And these are like really key components in animal cells. And then you also have these different receptors. You also have glycolipids, which are lipids with carbohydrates attached to them, but we'll talk about that later. For your glycoproteins, they face outward and they play a huge role in cell-to-cell recognition. Oh, I wish I had a red highlighter. I have orange, I guess that's close enough. And so here's your little glycoprotein chilling out up here. Ew, this is not orange. Okay, well, it lied to me. And so your glycoproteins, they just kind of stick out. It's protein with a carbohydrate. And when you look at the surface of the cell, if you were to like zoom in on this picture really close up, you'd see you have your like membrane protein kind of embedded, and then you have your carbohydrate sticking out like a little dingle finger. And so on your O type, you have just your standard carbohydrate group. On your A type, you have your standard carbohydrate group and then this little bonus. On your B type, you have your standard carbohydrate group and then a different little bonus. Now I need two other colors. Gosh darn it. Little bonus. Little bonus. And those little bonuses are additional sugars that are added in. For type A, it's acetylgalactosamine, and for type B, it's just galactose. And those little additional sugars are like make or break. So when your cells are going around and they're recognizing each other as your own cells versus a virus or an infection or a bacterial cell, you have your antibodies, and your antibodies, if you have these antigens that have specific sugars attached to them, are trained to look for sugars of the opposite blood type. So if you have A antigen on your blood cells, you have anti-B antibodies. That way, if you had some other type of blood in you, your body would know to go out and go attack it, which is great in most cases. In the case of blood donation, that's why you can't get blood from someone who doesn't have the same blood type. But this is helpful for your body in terms of keeping up your immune function and keeping you healthy. And in your cells being able to recognize some sort of intruder. And so these sugars play like a really, really key role. And so to write this up in like happy words and not Miss White rambly terms. Oh, where's red? There we go. Each blood type differs based on the sugars bonded to the protein. In the cell membrane. In cell membranes. So they all have, again, the default carbohydrate sticking out. For the glycoprotein, the glyco part is what indicates it's partially a carbohydrate, but then these additional types have these additional sugars. Why did I pick yellow? I don't have a yellow. Well, I have a yellow pen, but you can't see it. I guess I'll use orange. So type A has the regular group plus acetyl galactosamine type B has the regular group plus galactose. Type AB has both additional groups, or it has, I guess not both additional groups, but it has both types. Has both types on it. Like there's not a hybrid one that has both the acetylgalactosamine and the galactose. It just has both type A ones and type B ones on it. Does that make sense? I hope so. And type O, no additional sugar. Or like none of these at least. None that we know. I'll say that, because science is always evolving. I don't want to sound stupid in 10 years. And so for who can donate to a patient with group AB, so here's the little chart. I guess this would have been helpful when I was telling you about immunity. But so if you have antigen A, you have the acetylgalactosamine on your glycoproteins, you make anti-B antibodies. So your antibodies, they're like whatever to the acetylgalactosamine glycoproteins, but we will absolutely destroy any sort of galactose glycoproteins. And if you have blood type B, you have your glycoproteins with galactose, and then you have these anti-A antibodies that are like, we will absolutely destroy any acetylgalactosamine antigens, like those going downtown. If you have blood type AB, you have both the antigens with the galactose and the acetylgalactosamine, and then you have no like specialized antibodies. You have antibodies, like you're not like immunodeficient, but you don't have any specialized ones. If you have type O, you have none of the little specialized glycoproteins on you, and you have both anti-A and anti-B antibodies. So like your blood is going to go and attack any sort of antigen A or antigen B. So you really can't get blood from anybody but an O. If you're AB, you can get blood from pretty much anybody because you're not really going to attack anybody else's blood, which I know sounds horrible, but is actually really good because if you're ever in need of a blood donation, you're pretty receptive. Oh, duh. Okay, so here's the question. So who can donate blood to a patient with AB, A, B, AB, or O? They have no specialized antibodies. And then what group is called the universal donor? That is O. Because it doesn't have any antigens on it, neither of these specialized antibodies are going to go like absolutely sicko mode if O blood is introduced. They're just going to kind of be like, whatever, this is part of the team. Has no antigens to be detected by specialized antibodies. Smiley face. And then I think, am I now correct? I am now correct. Okay, cool. So that's it. Again, Monday, if you have any questions, please let me know. Hopefully this wasn't too painful.